In this rapidly evolving world, the only way for us to survive and thrive is to be adaptable to change, to keep up with the pace of progress, and to embrace new advancements and technologies. This has propelled Wyeth Nutrition with its firm commitment to disseminate scientific nutrition information to establish the Wyeth Nutrition Science Center as a credible nutrition science platform that supports healthcare professionals in nurturing healthier generations of Filipinos. The Wyeth Nutrition Science Center has been taking strides to develop relevant and accessible learning platforms that provide cutting-edge nutrition science information from major health and training institutions to renowned local and global nutrition experts. The Wyeth Nutrition Science Center equips healthcare professionals with the latest and relevant scientific information, supporting and empowering their professional growth through convergence, empowerment, connectivity, and access. Don't just keep pace with the changing world. Take the lead in nurturing healthier tomorrows with the Wyeth Nutrition Science Center.
Welcome back to the Wyeth Nutrition Science Center's virtual learning series. As doctors, we know how preterm infants are at an increased risk of delayed brain development. In my practice as a neonatologist, I have seen for myself just how much different the cognitive and behavioral outcomes are for preterm infants compared to how it is for infants born at full term. Today's discussion focuses on the brain development of preterm infants and its complications. We will look at impaired executive function and how it presents in practice, as well as the kinds of nutrition and other interventions that can ease the difficulties associated with disturbances of executive function. So let me introduce our speaker. Our speaker is a consultant neonatal pediatrician and professor of neonatal medicine, Newcastle University, Newcastle upon Tyne, UK. He leads a broad portfolio of translational research and is involved in numerous studies that include large-scale National Institutes for Health research collaborative studies. He is a member of the European Society of Pediatric Gastroenterology, Hepatology and Nutrition, Committee of Nutrition, and is the chair of the UK-based multidisciplinary neonatal nutrition network, as well as working with colleagues in Liverpool, UK, and Ibadan, Nigeria, helping to establish a neonatal nutrition network in sub-Saharan Africa. Friends, let's all welcome Professor Nick Embleton.
Hi, thank you very much for the introduction, Jojo. Lovely to meet you all, and thank you very much to the Science Centre for organising this webinar today. I'd like to thank all of the attendees, all of those of you listening in at the minute. I know how difficult things must be in clinical practice. We're facing similar challenges over here with the COVID pandemic. So thank you for finding the time to join the webinar today. I hope this is going to be a great platform to provide some good interactive discussion as well as the education. I look forward to getting your questions, and maybe one day in the future I'll be lucky enough to come and visit your wonderful country. So thank you very much again, Jojo and Bernie, for inviting me.
You're very welcome, Nick.
Thanks.
We're excited to watch your lecture this afternoon. And ladies and gentlemen, without further ado, the first part of Professor Nick Embleton's lecture, Preterm Brain Development and Developing Executive Function. Let's all watch this.
Okay, right. So thank you very much to Willie for the introduction. Thank you to Wyeth for inviting me. And just to kind of heads up at the start, I'm a clinician. I'm not a basic scientist. I still work on the neonatal unit. I still do all my own calls. So this is really the perspective of a clinician, how I understand some of this kind of complex science. So you won't see lots of complicated basic science graphs. This is a simplification as I see it from a clinical point of view. Some disclosures here. I have been funded by industry for some of our studies, but we've also had government funding for lots of our big trials. And really, this is the way that I think about how nutrition has changed for preterm babies over the last 10 or 20 years, that in the last 20 years, certainly, we've recognized the importance of nutrition right from the very beginning for our preterm babies, starting with parenteral nutrition, but also working with the mothers to help them express breast milk right from the very beginning. And undoubtedly, better nutrition is one of the key reasons why survival has improved in our babies. But as more of our babies survive, we're looking after more and more complex babies. We now look after quite a lot of babies born at 23 and 24 weeks. Many of these babies have lots of complications, necrotizing enterocolitis, and so we need to better understand how to provide nutrition to these babies. So as survival improves, the job of us to provide good nutrition gets even greater. This is my simplified form of brain development in humans, really emphasizing the point that we're quite different to most other primates. So most other primates deliver their young relatively mature. So most chimpanzees and monkeys, as soon as they're born, they can get out, they can hold onto the mother's fur, and they can crawl around. But a human baby is really very immature. A human baby just lies there on the bed and is unable to even crawl to the mother's breast. And the important point of this is then that when you have a premature baby, you miss out on the third trimester when this rapid period of brain growth is happening. And therefore, nutrient deficits in this time are likely to result in permanent neurological problems. Now, this is a Welsh cyclist, but we'll call him British because he won the Tour de France. And if any of you are cyclists, you'll know that in the last 10 years, British cyclists have won the Tour de France 5 or 6 times. It makes the French absolutely mad that we win it all. The Spanish as well, for any of you who are Spanish here, you haven't won it for many, many years. So this guy here, for 3 weeks, he cycles up and down mountains, 6 hours a day on a bike, up and down, up and down. And he weighs about 70 kilos. He has to eat 7,000 kilocalories a day to do this job. Just imagine how wonderful it is to eat 7,000 calories and to be this thin. That would just be amazing. So he has a huge energy intake, about 100 kilocalories per kilo a day to cycle up and down mountains. Put that in the context of a preterm baby, where we know that their caloric requirements are around about 120, 130 kilocalories a kilo a day. So our babies on the neonatal unit require an energy intake 20 to 30% higher than the most physically enduring activity known to man. That's really quite incredible. And when you think about what's using all that energy, unlike in adults, the brain uses 3 times as much energy per weight compared to an adult. So about 60 or 70% of all that energy we're giving to our preterm babies is being expended in the brain. And therefore, very easy to understand how a deficit of macronutrient intakes may impair brain development. So when we think about these different phases of brain development, certainly neuronal migration, very active in the 2nd and 3rd trimesters. Apoptosis, programmed cell death is a very important function. But then we need to start making all these connections. This is very active in the 3rd trimester and carries on throughout infancy. And myelination, as you heard, that may not be complete until you're 20 or 30 years of age. So there are lots of different processes. This is my simplified understanding. But what we now know is that lots of these processes in the preterm brain and presumably the term brain
are under the control of growth factors, and particularly IGF-1. And there's a lot of work now linking the levels of IGF-1 in preterm babies to the speed of brain growth and later outcomes. And we also know that IGF-1 is modulated particularly by macronutrient intake, so your intakes of energy and your intakes of protein. And so feeding babies the proper amount of macronutrients that they need is likely through lots of mechanisms to impact on brain development. So in humans, as I said, it's all about the brain. When I show this slide to my fellows, I normally offer to buy them a beer if they can find the brain inside the skull of the rhinoceros. So inside there, can you find where the brain is? So I'm not going to buy you a beer. I'm going to show you here that actually, although the rhinoceros has a very big head, the brain is actually really tiny. But in comparison, in humans, the brain fills the entire cranial cavity. And I know this to be true because this is my son. And of course, 90% of brain is genetic. Okay. Atul showed this graph yesterday showing that there's a direct relationship for most mammals between your size and your brain size. But in humans, we're off the scale here. And so relatively, our brains are about 10 or 20 times as big. And everything about placental development and nutrient transfer and the composition of breast milk and how we feed babies, everything is to do with the brain. Nothing about feeding was there to stop you becoming obese or developing diabetes. Those are all very important outcomes. But evolution was designing us to feed the brain. That is the primary reason for all the constituents that you have in breast milk and how the placenta works. So growth and differentiation is very complex, and particularly the third trimester is very active. And the brain volume increases by 3 mils every day. Your cortical volume goes up by a factor of 4 in the third trimester, and your cerebellar surface area goes up by a factor of 30. And we're only just starting to understand really how important the cerebellum is in terms of lots of these intellectual cognitive functions. So when you think about all these multiple anatomic regions and you think about all these unique developmental processes, all of these have to happen in sequence. So all that amazing data that you've seen today and yesterday, all these things have to happen in a programmed way. And you have to do step 1 before you can do step 2, before you can do step 3, etc. If you miss out some of those steps because you weren't providing the right nutrients, you can't go back to the beginning and start again. So everything has to happen in the right sequence. And if you don't provide nutrients at all these critical stages, you can't ever go back and repeat it. And we need to develop all these complex circuitry, all these connections that modulate the key behaviors and make us human as opposed to all the other animals on the planet. So this, for me, comes down to thinking about both the cells and the connections. It's not one nor the other. We need nutrients to do both. So when we think about this critical window, when I, as a neonatologist, and perhaps many of you, look after the babies from 24 weeks all the way through to 2 years of age, we recognize the complexity of all these nutritional processes. But then we recognize that we're dealing with lots of sick infants, lots of other things that may be impacting on brain development, plus the complexity of the environment in the NICU, all that machinery and all the other things we have to do. And then on top of that, we think about poor nutrition. And many of you will realize that the babies on the neonatal unit simply do not grow as they should do. So very good evidence that our nutrition is not good enough. When you put all these factors together, it's maybe not surprising that many preterm babies, when they grow up, have this slightly different cognitive or behavioral phenotype. It looks a little bit as if it has some features of autism. It's not autism. But when you think about it more, the features these babies have are impairments of executive function. And we're going to go on to explain some of this a bit. So I think about this in 2 ways. Partly this is due to damage. Well, this may be damage caused by a hypoxic insult or an IVH or something like that. But part of it is disordered development, that we simply didn't provide nutrients at the right stage to go through the normal sequence of developmental processes. This isn't purely simple damage and bleeding. This is lots of different processes that are being affected here. So the preterm phenotype has lots of different elements to it. If you subject kids to straightforward cognitive testing who are born preterm, on average at 25 weeks, you're about a standard deviation below the mean for your IQ or your developmental quotient. But then in addition, we see lots of features of impairments of executive planning. but also things like visuospatial problems and visuomotor integration. So this is not simply that preterm babies are less clever. They have deficits in certain domains. So we've talked a lot already about what executive function is, that this develops through infancy, childhood, and continues all the way through until adulthood. And we see deficits of this in preterm children, even when their IQ is normal. So they may have a normal IQ, but still be very impaired because of the problems with executive function. And not surprising, this is more common if you're a lower gestation or you have more complications. But importantly, this is modifiable. So a lot of data showing that the home environment or the school environment has a major role to play in executive function. And Atul was talking yesterday that this isn't simply just nutrients involved here. This is how we play with our children as well. So we need to think of nutrition in a very holistic sense, not get too obsessed with single nutrients. Apologies to all the biologists there, but to think about how we look after babies in the home environment. Okay, so these are the three elements that Pascal talked about earlier: working memory, cognitive flexibility, and inhibitory control. And this is how it applied to me when I had the invite. So first of all, I had to remember what time the plane was going to depart and how to get from the airport to the hotel. I had to sort of hold that memory in there. Cognitive flexibility, this was how could I make the talk interesting to those of you here who are maybe more basic science, as well as clinicians. So I'm thinking about the same problem from two different aspects. And then as we talked about inhibitory control, how did I manage to finish my talk before I got here without constantly being distracted by my emails or watching the television? So all these things go together to make up executive function. And here are some of the key skills. This is paying attention, as hopefully you're doing now, organizing and planning, initiating tasks and staying focused, regulating our emotions. I'm sure many of you are very excited, but you're doing a good job to keep calm now, and self-monitoring, keeping track of where we are. And then this relates to some other skills, people what they call kind of hot executive function. This is to stay on task when something else comes along that might be more exciting and resist the temptation. But also within that, the ability to reflect, to sometimes just stop and pause and think, should I be doing things a little bit differently? And then being able to do this reflection quick enough that you can stay on task. And when I was developing this talk, I thought this is quite similar to how we practice newborn resuscitation. So many of you are pediatricians here, and all of you at some point in time would have been involved in resuscitating babies, and you'd have followed this algorithm here that tells you how to kind of check the breathing and check the heart rate. So all of us have to retain this algorithm in our head and go through it. But when things aren't quite working out, we need to recognize not to just keep on plowing down there. You need to be able to stop, think laterally and think, so why is the kid still blue? And maybe my tube's blocked. Maybe it's in the wrong place. These sort of things. Check those things, but then go back to the task. So being able to perform good neonatal resuscitation requires a very high level of executive function. And I think all of you can see how you might be incredibly clever, but without good executive function, you couldn't do some of the day-to-day tasks that we perform as clinicians here. So how is this manifest in some of our kids? So thinking about the preschool environment, kids with poor executive function, they typically get more frustrated. They're more likely to throw things around. They won't ask for help. They maybe kind of kick out or scream rather than really being able to work through the problem. More likely to have temper tantrums, more likely to be quite fixated and wanting to do things in the same way each time. They don't like change. And then when these kids get to school, they maybe start on one thing, but then they get distracted. They move to another task. They get all their assignments and their homework mixed up. They couldn't remember which one they were doing today. And they tend to panic when rules change. So when on Tuesday you don't have your regular sports activity, that's shifted to a different day. They also find it harder. They take longer to complete their tasks. Their processing speed is slower. So they can't quite finish the assignments. They can't finish their exams in time. And they tend to be a bit more impulsive and not so good at working together. So we see these features quite a lot in our preterm babies. So just as a recap, human brain development is very complex and rapid in the third trimester. You need lots of different nutrients there. And this is about cells and connections together. And the executive function is integrating all these kind of complex tasks to allow us to have goal-directed behavior, to regulate our emotions. And it manifests in different ways. So the way that it would present in infancy at a 3 or 4-year-old is quite different to how it presents at 9 or 10 or when you're an adult. And this is a major issue for lots of our babies. So this is data from a very important study in the UK, the Epicure study, which just looked at very preterm babies less than 26 weeks. And they saw them again here at 11 years of age. And so they had 161 very preterm babies, and they compared them to matched controls. And they looked at a whole set of outcomes: IQ, math skills, reading, executive function, visual processing, and sensory motor skills. And what they described was what they call comorbidity. And that's where you have a problem in more than one domain. So you may have a low IQ along with executive function, or you may have executive function problems and sensory motor skills. So comorbidity means more than one domain that's causing a problem. And what they found in this cohort of less than 26 weeks, around about a half had some impairment of intellect or learning, compared to just 5% of the control population. But importantly, about a quarter had comorbidity. And in the control group, none of the control kids had any comorbidities. If they had a problem, it was only in one domain. So a quarter of our preterm babies have problems in more than one area, and this really adversely impacted on their ability to learn. And particularly, they found difficulties with maths. Classically, these kids find maths really, really difficult. And the maths problems, particularly associated with working memory. You've got to remember things in your head, how to do your equations, but also, say, other aspects of executive function and visual spatial processing. So are we likely to see these problems in adults as well? Yes, I think we're just starting to see some of these problems coming through in adults. And this was a study from a couple of years back, just looking at young adults and looking at brain size, and they were able to test some of these adults with cognitive tests, the Wechsler, but they also did MRIs, and they were able to look at different brain regions, and they could show that brain regions, so particularly the thalamus and the corpus callosum, in adults who were born preterm were smaller than controls. They had slightly larger lateral ventricles. And all of these problems were associated with all the factors you would expect, so associated with lower gestations and more neonatal problems. But they were also able to relate brain size to some of these measures of IQ in later life as well. And this suggests that insults, so damage or disorder development from very early life, are likely to track through into adulthood for lots of our babies. And they were able to rate, as I say, IQ to some of these measures. So in green are the control dots, and in purple here are the preterm kids. So this is total intracranial volume, gray matter and white matter, and this is full-scale IQ. And you can see that for both term and preterm adults, born adults, there's a relationship between brain size and IQ function. But what you see is that for the very low birth weight preterm babies, that the whole thing is shifted down on an average. This is shifted down about 1 standard deviation. So this is a great review. Raju here, you can pick up the references later on. This is a lovely review of a whole range of outcomes in adults who are born preterm. So he looked at sort of metabolic outcomes as well as cognitive outcomes and psychiatric outcomes. It's a great review. And he noted in this that lots of the adults who are born preterm had higher rates of anxiety and depression. They're more likely to have had school problems. They particularly struggled when they transitioned from being a child to being an adolescent, a very challenging period for these kids. They were more likely to display withdrawal or autistic behaviors, and they were more likely to report being bullied at school as well. And boys and men tended to fare worse than girls and women. And what they describe is that adults born preterm had different, not necessarily abnormal, phenotypes and personality types. They're just a little bit different, some of our preterm kids, as they grow up. Not necessarily purely abnormal, but there's something about the preterm behavioral phenotype. There's maybe a little bit of comfort here that they reported less negative emotions than their peers. They tended to be a bit more cautious and shy, and therefore they tended to be much less likely to be involved in risk-taking behaviors, less likely to be involved in smoking or drug misuse, teenage pregnancy, and all these kind of things as well. They're just generally a little bit less adventurous. So this is a great study that's tracked a cohort of very preterm babies in Bavaria through until adult life. So quite a decent size, 260 preterm babies. And they measured attention at 6 years of age, 8 years of age, and 26 years of age. So a fantastic longitudinal cohort. And what they showed was that preterm kids and adults have more attention problems. They were more likely to have a diagnosis of attention deficit hyperactivity disorder. And although these problems get better as they get older, they still persist into adulthood. So this is some of the data from that study. Just to orientate you, on the right here is females. On the left here is males. This is the measured outcomes at 6 years, 8 years, and 26 years of age. And the top here, these are parent-reported attention problems. And the bottom here are the observer measured by the psychologist. So if we just concentrate on the boys here, what you see is that in red are the preterm boys, and in blue the term boys. And so in terms of how the parents rate attention problems, preterm babies are rated as at 6 years of age of having many more attention problems than term. Gets a little bit better at 8 years of age, but you see here, even at 26 years of age, the parents are recognizing attention problems in the adults who are born preterm. Here the scale works the other way, but you see the same feature, that compared to term babies, preterm babies have shorter attention spans at 6, 8, and still at 26 years of age. So although they catch up, they're still delayed at 26 years of age.
So, Professor Nick, are you ready to answer questions?
I'm ready. Fire away.
Great. Which particular part of the brain is involved in executive function?
Yeah, that's a great question. Clearly, brain development in humans is incredibly complex. And I think I outlined some of the challenges that preterm babies will face. The MRI studies in preterm babies do show differences in certain parts of the brain that appear to have maybe grown a bit more slowly. And there is a bit of work linking those areas to executive function, perhaps. People particularly interested in the fibers that cross the corpus callosum, but also the basal ganglia. There is a bit of work showing that poor nutrition in preterm babies particularly affects the size of the caudate nucleus, or the basal ganglia. But more recently, there's been quite a lot of work looking at the cerebellum. And I think in the past, as neonatologists, we tended to sort of ignore the cerebellum, but we're now really recognizing the importance of cerebellar growth in the third trimester. We note that in lots of preterm babies, that appears a bit smaller. And I think that's probably also related to impairments of executive function. But I think the key point is that brain development is clearly very complex. You need lots of different brain regions. You don't just need the brain regions themselves to develop. You have to develop the right connections between these areas in order for you to have good executive function in later life.
So I guess it doesn't, it's not related to the vulnerability of the germinal matrix?
No, I don't think that's been particularly identified. I mean, no doubt if you have intraventricular hemorrhage, you might damage brain parenchyma and brain development in other ways. So yeah, that's clearly important, but that isn't something that's come out in the literature. I think one of the key bits about executive function is that it does seem to be susceptible to overall brain growth, and particularly insults from poor nutrition. So I think probably germinal matrix hemorrhage is probably not so relevant in this aspect.
Okay. Although I did see literature which describes decreased cerebral white matter, particularly frontal, parietal, and temporal.
Yeah.
But you're saying that the cerebellum is very important in executive function?
Yeah, I think you need you need your cerebellum to provide this continuity of tasks. And I think certainly in my learning when I was a medical student, you learn about the cerebellum and you think of it purely in terms of motor development. But we're now recognizing that you need your cerebellum to enable all your thoughts and process to continue in a kind of smooth fashion. And we do see, for example, challenges and impairments of communication and speech development in association with the cerebellum. So yeah, I think the key message is that brain development is incredibly complex, and we shouldn't get focused on a single region. We need to think about the totality of nutrient provision for brain development.
Okay, one last question from me is it seems that the more premature, the higher the risk of a deficiency in executive function. Can you comment on the incidence of poor executive function among the late preterms, those who are 34 to 36 weeks, maybe?
Yeah, that's a great question. And we tend as neonatologists to get very focused on the very preterm babies. Most of the work is in babies sub 1,500 grams, 32 weeks, and they're the babies who've ended up in most of the studies where I've presented the data. But yeah, it's a great point. Babies born late preterm, 34 to 36 weeks, also have differences in executive function, behavioral development, and these things. Again, the mechanisms are probably in part due to nutrients, but maybe due to other factors as well. I suspect the impact is less severe than in a 25-week baby. But you're absolutely right to highlight that as pediatricians, neonatologists, we really do need to be interested in these late preterm babies as well, because they're also at risk of later life problems.
Yeah, great. I've been told that we have live questions.
Okay.
From the chat box, from the Q&A box. Okay, yeah. The first is in the Epicure studies, were the preterm babies devoid of any structural abnormalities, such as intracranial bleeding?
Yeah, important question. My understanding is that the follow-up studies provide data on all the babies who were recruited. So these included all babies less than 26 weeks in a single year in the UK. And so a number of them will have had interventricular hemorrhage and other structural brain problems. I'm not sure it's necessarily helpful to separate those babies out. I mean, clearly babies with a grade 3, grade 4 hemorrhage are likely to have a worse cognitive outcome. But really what we're trying to emphasize here is the vulnerability of all of these babies. Even babies who have a normal ultrasound scan or a normal MRI scan are still at risk of later life problems. And one of the things that we're able to do perhaps is pay closer attention to nutrition, nutrient intakes in early life. That's one thing that we can do something about. It's actually quite difficult to prevent these babies having hemorrhage, but nutrition is something that we can work on together.
Great. The second question: does prematurity affect? This is a question on intelligence quotient.
Yeah.
Not not necessarily executive function. So
Sure.
Does prematurity affect the IQ of the infant?
Yeah. I mean, I think that there's really quite a lot of data showing that preterm babies have lower IQ on average in later life. Clearly, lots of preterm babies, even babies born at 23, 24 weeks, go on to have normal IQ. So it's very important to recognize that most of the babies we're looking after end up with an IQ in the normal range. The majority of our children go to normal schools. It is true that as a group of babies, they tend to have a slightly lower IQ than their peers born at term. But for any individual is able to achieve great things. So we shouldn't automatically assume that all the babies are going to have impairments of either executive function or IQ. Lots of these babies do absolutely fine.
Okay. The next question: what could be the cause behind differences among males and females?
Yeah, that's a really interesting area of work, and there are groups working on this, particularly a group in New Zealand looking at differences between boys and girls in later outcome. Some of this may result or may reflect slight differences in nutrient requirements. We're not really quite sure whether boys and girls need different diets. I don't think we've got enough data to say that, but it's clear that later life outcomes and metabolic outcomes do differ between boys and girls. And as I say, that might relate a bit to nutrient requirements, but also might relate to a whole series of endocrine hormonal factors that modulate some of these differences. So I'm not aware that girls necessarily are at less risk of impairments of executive function. I think we should consider that all the babies we're looking after are at risk. There may be slight differences, but I don't think that has any practical relevance for how we look after babies on the neonatal unit.
Okay. Next, what is the difference in the effect of breastfeeding versus formula feeding on cognition?
Yeah, I mean, that's another great question, a really important point here. the more research that people do, the stronger the data becomes supporting the benefits of breast milk. I don't want to get into a debate about donor breast milk, but what we know is that fresh breast milk from the mother is by far the most important nutritional intervention. And there's very strong data showing that later life outcomes, be they of IQ, executive functioning, measures of brain growth, white matter development, are strongly linked to the amount of mother's breast milk that you get in early life. How does it work? We don't really know that yet. There are so many components in breast milk that may be modulating those differences. Clearly, there are some aspects of fatty acid composition, but also other components, sphingolipids, sphingomyelin, phospholipids, all of these important precursors of metabolites in the brain and cell development in the brain. So, yeah, there are important differences. We're absolutely sure that these reflect something about the biology of breast milk from the mother. And therefore, as I say, like the key learning point always in all these things is, whilst we are very interested in the biology of all of this, the one thing that all of us can do is help support mums to provide their own breast milk as well as they can.
And now let's continue the WNSC virtual learning series with the second part of Professor Nick Embleton's presentation. Let's watch this.
So, how are the various ways that we can test for impairments of executive function in the clinic or in some of the research studies? And really the point to make here is that if you really want to test executive function, you need an expert. So this is kind of beyond me as a pediatrician. I would need to work with a psychologist to really perform these tests properly. But I want to just give you an idea of how it can be done. We can supplement some of these more expert tests by using questionnaires or simply observing the kids or listening to what the parents say. So say these tests, there are lots and lots of them. They're age specific, but you've got to use a validated test. You need expert support if you're going to do this. As I say, there's some questionnaires. The brief is a questionnaire, CBCL is another one, and then there are some more formal tests to do. But just a kind of example of the sorts of things that you might test. So a very simple one is just to track visuomotor tracking skills, and this you would get a kid with a pointer on a computer screen to follow this little red dot. So the kid has to follow it round, and the computer can measure how good you are at tracking this ball. Another test would be this short-term memory function. So what you do is you get a kid, you put a set of headphones on him, and you get him to watch a computer screen. And then some images will come up, and you might show him some pictures, one of which might be a hat. But also in his ears, he might be hearing different words, and he might hear the word hat, but not necessarily see it. And then the operator here would ask the child, have you seen this hat before? Have you seen the car before? Or did you hear the word hat? So this is trying to test short-term memory. So that's another sort of test. Pascal talked about this before. The sequencing test, you might show kids different size shapes and you get them to sort them out, or you might give them a list of numbers and trying to sort those numbers quickly into the right order. This is trying to get tests doing two things at the same time. So on the computer screen, the words of different colors will come up, so red and green, but where the word comes up in the correct color, then the kid has to click the button. So they're concentrating that, but then they confuse them by getting them to follow this white dot as they're doing it. So they're having to follow this white dot and think about these things at the same time. So can they do both of these activities at the same time? And then a recognition test, show the kid 3 different objects in sequence. So a dog, a bat, and then a key, and then show them a series of pictures and get them to click when it comes up in the right order. Can they remember that order and then click when the correct one comes up? We use this, the CBCL. This is quite an easy thing for parents to complete, where they rate different skills as 0, 1, or 2. So these are some questions that you would find on this form. Can't sit well, restless or hyperactive. Demands must be met immediately, easily frustrated, doesn't want to go out of the house. And then parents will rate these kids 0, 1, or 2, and then you can add them all up and give the kids a global score. So we've used this in some of our studies. So I'm just going to show you a few case studies. These are from kids who I've actually looked after in my clinic and still in contact with. They're all anonymized by names. So the first one is Moya, who was born at 23 weeks. She had NEC. She's now 9. Physically, she's actually pretty normal. She goes to a normal school, but she's very much in the kind of slower half, the bottom of the class. She's really, really poor at maths, just can't do maths. But otherwise, the teachers say, she's okay at other things. But at break time, she tends to play with the boys. She tends to play football and running around games. She can't do the make-believe. So she can't play mummies and daddies with her girlfriends. She can't, she doesn't play with dolls and these sorts of things. So her activities and her ability then to interact with her peer group is quite limited. And if you ask her mum, she says she actually hasn't got very many good friends. So plenty of kids to play with, but no particularly close friends because she just hasn't got this complexity of higher thinking. The next is a little 24-week boy who was a twin. Sadly, his twin died at 2 weeks of age. And those of you working on NICU recognize this is a particularly common and difficult problem. He's now 7 again, physically pretty normal. He's incredibly hypermobile, very, very flexible. I don't know whether that's anything to do with it. And his speech is really very, very poor still. So you have to really struggle to kind of understand him. Again, he goes to a normal school, but is in a special needs class. He's amazing at kickboxing. He loves slapstick comedy. So really simple comedy, falling over, cream pie in the face and these things. He finds it absolutely hilarious. But he doesn't understand any more kind of complex humor. But actually, he's a very, very likable, sociable boy. So when he comes to clinic, it's a real pleasure seeing him. I love seeing him. But you can see he's never going to be able to live on his own. He's never going to have an independent life. He simply hasn't got any executive function that would allow him to leave the house and go to the shops. He just couldn't do it. And the last one is a boy who was born very prematurely. He had a small IVH when home and oxygen. He's now 24. Hasn't got any medical problems at all. He went to college and he learned cookery schools. He now works in a school canteen. He loves his routine, but he still lives at home with his parents. He tends not to go out at all. He can't really function in big groups. So he's great in the kitchen on his own, but he can't work with others. Really got very few friends, but if you ask him, he says he's happy. But he comes home and he will spend 6 hours on the PlayStation. And he's great at these kind of computer games. So he just doesn't really interact with anybody else. But if you ask him, he feels his life is fine. He hasn't got an issue with it. So those are some case studies. So why does that happen? So say we need to think both about things that might damage the brain, but also things that might sort of impair the normal developmental processes. And there are lots of different factors that might involve damaging the brain. So the cytokine storm associated with preterm labor, maybe some genetics, hypoxia, toxic exposure, maybe some environmental toxins on the NICU, but also obviously things like bleeding and hemorrhage. So these are all the things that might damage the brain. Then all the factors that may confuse normal brain development. So all that noise and light on the NICU, all the pain, all the sedatives and drugs we give to control that, all of the sort of disturbances are caused by inflammatory episodes, but particularly, as I say, poor nutrient supply, and maybe disturbance of gut-brain axis and lots of things. So lots of different reasons why we may see these problems in our babies. And if you go back to this kind of simplified schema that I had before, you could think about, well, maybe migration, maybe these cells just aren't quite migrating in the correct fashion in the third trimester. They're getting different exposures to what they would get in utero. Or maybe there are fewer neurons there. Maybe the programming of apoptosis, or maybe later on the programming of synaptic pruning just doesn't happen properly. Maybe the cells aren't quite dying off when they should do, or maybe the wrong types of cells are dying off at the wrong time. Maybe they're just not making enough connections, or they're not making enough connections at the critical time points. And then maybe myelination just isn't happening. And I think you've seen a lot of data show that this processing speed from not having enough myelin may be impacting on some of these measures. So this is both grey and white matter. So how do you make it better? Well, as I say, this is incredibly common in lots of our very preterm babies. It's complex to assess. You need age-specific validated tests. And the impact of this is going to change throughout the life course. This is going to present differently, obviously, in infants and young children to how it may present in adulthood. And you've seen a lot of data showing the neuroimaging correlates to this. So what can we do about it as pediatricians in the clinic? Well, of course, we need to kind of recognize that it's a problem and understand this has a huge impact on family lives. So as pediatricians and neonatologists, we've tended to document cerebral palsy as an outcome in lots of our studies. My experience is that kids who just have a hemiplegia but no neurobehavioral problems actually function incredibly well in later life. But you can be the most physically adept child, but without executive function, the impact on the family is enormous. So this is really a big problem. When I say we're only starting to realize it now, because 20, 30 years ago, most of these babies didn't survive. So we need to recognize and refer for support, but we also need to think more about what it is we're doing on the NICU when babies are first born. We need to be thinking about their brains, how we stabilize them, how we control oxygen, glucose, and really fine tune everything that we can. So nutrition is a big thing. So how might nutrients impact? So this is just a kind of schematic idea. This isn't based on lots of data. But obviously the impact of a nutrient will depend on the amount of that nutrient that's lacking, the type of that nutrient, what's it involved in, the timing and the duration. So a short insult that's very severe, that only lasts for a few weeks, will have a very different impact to a slightly lesser nutrient deficiency, but one that goes on for a much longer period of time. So these are just some sort of theoretical ideas. You might have something here which has a really big impact when the babies are first born, but after 1 or 2 weeks, maybe we get better at providing those nutrients. Or you might have a nutrient that isn't so important when you're first born, but becomes very important when these babies start to grow. Or there might be a nutrient that isn't lacking so much at the beginning, but all the way through the first 1 or 2 years of life, we're just not providing enough. So examples of this might be macronutrients. We know if you go back to your units and you look at how many macronutrient protein and energy you give these babies in the first few days, you will find that most of the time you're simply not giving enough protein and energy to get these babies to grow. Once they're healthy and you can fill them up with lots of fortified milk, well, you meet the macronutrient intake there. But often macronutrient intakes are suboptimal in the first 1 or 2 weeks. IM might look a little bit like this. In the first 1 or 2 weeks, although you're taking lots of blood samples from the babies get transfusions. So there's plenty of iron mopping around in this first 1 or 2 weeks. But then when the baby is just on breast milk and not receiving iron supplements, they're very likely to become iron deficient. But when you go home, we remember to give them iron, so we all start them on iron at discharge. So iron might be having an impact at a different point in time to macronutrients. And then something like choline, DHA. We know, high amounts of accretion in the 3rd trimester, but lots of our babies won't get anywhere near enough choline, DHA in their intravenous lipid solutions. Even the amounts in breast milk probably aren't enough to match 3rd trimester accretion. And then when these babies go home, they may not be getting enough choline and DHA in their diets. So how nutrients impact on executive function is going to be very difficult to disentangle. So what are the different ways that nutrition may impact? So I can think of at least 5 different mechanisms. The first is, of course, you need nutrients to build your tissue. So simply not giving enough macro and micronutrients, you just can't build your brain tissue if you're not providing the substrate. The next one is you need energy to drive the system. Remember I said like 70% of your energy expenditure is in the brain. If you're not giving enough carbohydrate and lipid, you have to burn your protein for energy, and then you can't use it to build tissue. So we need energy to drive the system. Nutrients are important because they contain or they promote growth factors. So we know lots of your branched chain amino acids particularly stimulate insulin and IGF-1. So key nutrients involved in signaling to the brain. Some of the nutrients might be involved in gene expression, so various epigenetic processes, particularly B12, folate, betaine, choline. These things are probably involved in that. And then the last one would be preventing disease. So NEC is devastating for the brain. The cytokine storm kills off all your oligodendrocytes. So using nutrition to prevent disease is probably the 5th mechanism by which nutrients may impact on brain development and executive function. So this is a picture from my neonatal unit a few months ago now. And just look at the complexity. Here's the baby here. He's on a brain monitor. He's on a ventilator. He's getting nitric oxide. He's got cardiovascular monitors around him, syringe pumps. We took an X-ray. That's what neonatal intensive care looks like. It's really, really complex. But in all of that mix, people aren't seeing the nutrition. So there's nothing there really that people are doing at this point in time that is done purely with the aim of giving nutrients. We're trying to fine tune the oxygen saturations, the blood pressure, and all these other kind of things. And this is what we talk about on the ward round every day. You know, what ventilator mode should we be on? You know, sys control or pressure support or time cycle, pressure limited. I mean, all these different things. CPAP or high flow, or do we leave him on the ventilator? His bilirubin's 240. Should I put him on phototherapy? Should I stop it? Should I put some central lines in? How much fluid should I give him today? His blood pressure is a bit low. Which inotrope do I use? Should his saturations be 85, 90, 95? What's the low blood pressure? How many days am I giving antibiotics for? 2 days, 3 days, 4 days? Shall I repeat the full blood count? Shall I repeat the CRP again? When am I next going to check his blood gases? This is what we talk about all day long on the neonatal unit. And in all of that, nobody's asking a question about nutrition. It's completely invisible. So none of you can see that you're busy malnourishing this baby very successfully for a few days at a critical stage of brain development, because nobody yet has designed a little machine that alarms if you're not making nutrient intakes. So if you're not providing just enough oxygen, the big alarm goes off and you run over there and you give them a little bit more oxygen. Or the blood pressure drops a bit low, the nurse comes find you, you measure it again, you put them on some inotrope. So we fine tune the cardiovascular system, the respiratory system all day long. But nobody's fine tuning the nutritional system. People just aren't thinking about it because you can't see what's happening. And I think that's the biggest challenge for nutrition. It's just not there in front of you. And so when all the residents and fellows come to the neonatal unit, they're all terrified. Can I get the tube in? Oh, I can't get an IV in. And what fluid should I give? And when the consultant comes on the ward round, is he going to ask me for the blood gas? I better do another bite. This is what they're thinking about. But nobody's thinking about nutrition and the complexity here. So what's the problem here? The problem is you don't know what to give. That's sorted out by education and guidelines and meetings like this. You don't know what you're doing. Well, we need to audit our outcomes and we need to provide better leadership. People don't focus on nutrition, so we need to promote the importance of this on the neonatal unit. We're not very good at promoting breastfeeding in the first few days. And we need more research to obviously give us the data to know what to do. So what are the solutions here? Start nutritional management from the minute the baby comes onto the unit. Think about parenteral nutrition. Think about supporting the mother to provide breast milk. Conduct regular audit, educate, teach, learn, read, and read again. I spend my whole life reading. I must read 10, 20 papers a week. And you have to keep on doing it, and you have to teach yourself rather than expecting somebody else is going to come along AAP and give you a guideline as to what you should do. We should be thinking this for ourselves in our own units. And we need to provide leadership. So if you guys don't talk about nutrition, your fellows won't think it's important, your residents won't think it's important, your nurses won't think it's important. So we talk about nutrition on the ward round and we talk about breast milk. We have our mothers present when we do the ward round. And I make a point of talking about what a great job she's done to provide breast milk for that baby today. She just brought in a few drops, but I talk about it. And if me as the consultant emphasizes the importance of that breast milk, the mum feels really inspired. So unfortunately, we have a huge role to play in providing leadership in these situations. And then most importantly, we have to believe we can make a difference. If you think, well, these are sick babies, I can't get good PN, all that kind of stuff, I can't do anything, you won't make it better. I guarantee you, if you want to make something better, if you really believe that you have the power to improve nutrition in these babies, it will get better tomorrow. So you have to believe that it's within the grasp of all of us as clinicians to do a better job than we're doing at the minute. But if you don't believe that, it isn't going to happen. So lots of different interventions. So we've talked a lot about breast milk and IQ and microbiome, importance of meeting macronutrients, iron, and I'm just going to finish off with choline. I mean, choline was something I'd never even heard of before I started doing nutrition. I mean, what on earth is choline? I mean, I've heard about amino acids and fats and vitamins, but nobody ever taught me about choline. And choline is this sort of compound, a bit like a B vitamin. You find it in sort of meat products, fish, eggs, Brussels, apparently. Obviously, you get lots of it in breast milk, not surprisingly. So some recommendations have come up with. People reckon that adults need about 500 milligrams. Theoretically, very, very high intakes. You might cause problems through all the sort of metabolites that are produced. And you, not surprisingly, need higher intakes during pregnancy and lactation, very important for brain and fetal development. A lack of choline actually results in a similar phenotype to folate deficiency. And there's a bit of evidence that in adults, adequate choline may prevent some of your cardiovascular diseases as well. So a lot of animal work showing that you have a very high demand during the third trimester involved in fetal and placental growth. But again, it's one of these nutrients very involved in methylation and other epigenetic processes. And as a precursor for acetylcholine, not surprising, it's involved in neuronal proliferation, migration, maturation, plasticity, synapses, and all these kind of things. And when you look at preterm babies, often their intakes of choline are quite low. So maybe this is an important nutrient for us to think about. So there's been some rat models showing that supplementing mother rats improves spatial memory and learning and reduces cognitive decline. So there are going to be lots of mechanisms here. But choline, as I sort of said with the other nutrients, it's involved in substrate formation. It's going to be involved in signaling. It's going to be involved in genetic processes. So lots of different ways in which choline might be important. And this study came out last year. It's the kind of first study I think I've seen where a single nutrient has made a difference to a brain outcome in an otherwise healthy population. So these were healthy pregnant women delivering at term who recruited at the beginning of the third trimester to a trial where either they got the standard intake 480 or they had twice the standard intake. And this was a really complex study. There's only 24 mums in it, but they basically made the meals up for the mums. The mums ate pre-prepared food. I think sometimes that'd be quite a nice study to be part of. I wouldn't have to cook at all. So what they did was they're able to know exactly how much choline these mums were getting. So these were term healthy babies. And then they tested spatial and other sort of memory functions in infancy and showed significantly faster processing speed in the term healthy children whose mothers had took higher choline intakes during pregnancy. And when they looked at infants getting the lower amount, they also showed that infants getting the decent 480 milligram intake for longer during pregnancy also had outcomes. So some evidence of a dose response as well as an impact of supplementation. So I think this is quite an important, interesting study. Clearly, we need a lot more work. But if you can show this effect in completely healthy breastfed term babies, imagine what we might be able to do with preterm babies who we know are likely to be very deficient in choline intake as well as all the other things. So I think a lot of hope that we might be able to do some good trials. So this is a summary, remembering that there are lots of different ways nutrients may impact substrate, fuel, signaling, gene expression, or disease, that the impact of that on the later phenotype is going to depend on when the insult happens and how severe it is and how long it lasts for. And all of this is going to impact on executive function, which we can't measure in humans in the first 2 or 3 years of life very well. We're going to be needing to do longer term studies to pick these things up. So take-home messages, executive function complex, changes throughout the life course, increasingly recognized as an important problem in adults who are born preterm, complex etiology, but really emphasizing this is both cells and connections. That deficient nutrient intakes remain very common. We need to focus more, better audit, better QI, better leadership from all of us. We need to really focus on supporting breast milk for these mothers on the neonatal unit. This is really hard work for them to manually express for all those weeks and to be motivated to do it when they're really worried about their babies. And we need better trials, so support research, but I think a lot of hope in the future that we're starting to get an idea of the sorts of interventions that we could test in trials. And just a final plug, we've got a big research group and we've got a website, neonatalresearch.net. So all the slides from today and all the slides from all the other talks and presentations we do are all there. all free to view to the public, but also you can see some of the big studies that we're doing and the other work. And with that, I'm going to finish. So thank you very much.
Thank you, Professor Nick. That was a quite fascinating second part of your lecture. If we could only give you a virtual applause.
Thank you.
I have a question myself. You had mentioned, especially in the first part of your lecture, that the home and school environment is very important. As a neonatologist, I'm guilty of not referring to developmental pediatricians unless I see developmental delay. Is this an argument for early referral, perhaps for early intervention, regardless of whether there is a clear developmental delay among the preterm patients?
It's a great question. I guess it depends a bit on the kind of resources that you have available in your own healthcare setting. I mean, clearly, we would always want as much input and support for premature babies as we can get, but we always realize that resources are limited. I think your point about the home environment is really, really important. I think that actually one of the things that we should be doing as neonatologists is encouraging parents that they have the ability to affect some of these outcomes by the way that they play and interact with their children. We're all very aware that lots of kids, including my own, spend way too much time sitting on iPads and TV screens, and that they're not interacting, and you need to have this interaction for you to develop some of these executive function skills. So it's not like you need to have therapy or you need a specialist to tell you how to do these things. We need to be promoting normal child-family interactions, behaviors in the home, in the school. And really, I think I would say that's, for me, a far more important aim than maybe seeing a developmental pediatrician. Clearly, if you have major impairment, you need a developmental pediatrician. But I think all of our kids, we need to be emphasizing to parents that they need to be creating normal home environments that don't involve you just interacting with the screen.
Right, great. I guess screen time is a universal problem, even in the Philippines.
Absolutely. Yeah, just as much here.
All right. As a neonatologist, we have been very conscious about giving proteins early. We have been told that could be a cause for extrauterine growth restriction, and deferring proteins at birth can actually lead to lower brain volumes. Do you think that proteins might be the most important nutrient that should be supplied early on in preterms?
I would say that if people want to focus on one or two things in terms of nutrient delivery, one is macronutrients, so both protein and energy. I mean, there's no point giving lots of protein if you're not going to give the energy to accrete that protein into tissue. So you need both macronutrients. Of course, micronutrients are important, but for me, if I was to tell people to focus on two things, one would be macronutrients and the others would be maternal breast milk. Everything we know about maternal breast milk improves every morbidity and every outcome you can measure in premature babies. So for me, if you just want to focus on two areas, it's got to be mum's breast milk and macronutrients.
Speaking of mom's breast milk, there was a question on banked breast milk versus mother's or fresh breast milk. Could you tell us the differences between those?
Yeah, I mean, again, a very, very good question, a very important topic. Lots of us now recognize that providing banked donor breast milk may be a better alternative to formula when we're first starting to feed babies. It does seem to be associated with lower rates of NEC and maybe sepsis. So there's probably advantages to donor milk at that stage. But the studies have not shown any long-term cognitive benefit from using donor milk as opposed to formula. So early on, I think we're clear we would prefer to have banked breast milk. Later on, maybe it doesn't matter so much. But the important point, as you're trying to say, is that banked donor milk is not the same as maternal fresh breast milk. Following pasteurization, lots of things have changed. lots of the functional proteins are preserved, so you still see very high levels of lactoferrin and those sort of proteins, but you see much lower levels of many kind of hormones, growth factors, enzymes. Many of these destroyed by pasteurization. You also don't have any microbiome associated with it. We're very interested in the microbiome in our research group, and of course donor breast milk is pasteurized and has no microbes in it either, unlike mum's milk, which is teeming with bacteria. So yes, donor milk is a second best alternative, but really our efforts should be focused on trying to get as much from the mothers to their own baby as we can.
Thank you for that answer. Really useful even for my practice. I was told that there are now questions being submitted by Slido, so let's go over these questions on the screen. So the first one is regarding nutrition for admitted babies at the NICU. In your practice, when do you start with TPN?
So I'm going to talk just about preterm babies here, but assuming we're talking about a baby less than 32 weeks, we start parenteral nutrition on admission. The baby gets admitted to the unit, we put an IV in or an umbilical line, and we will connect that baby to parenteral nutrition, both aqueous and lipid, at hour 0. We give around about 1.5 grams of protein and some 1 to 2 grams of lipid per kilo a day. And we start micronutrients and everything else at that stage. So we started on admission, but we don't start at a full amount. I think it's important that we have time to reassure ourselves the babies are not acutely sick, because if you're a very sick septic baby, you cannot tolerate high amounts of nutrients. So I think that first 24 hours allows me to work out if the baby is stable, and in which case I would increase the parenteral nutrition on day 1, day 2. If the baby is very sick, I might stick at the starting amount. But I believe it's safe to start around about 1.5 to 2 grams protein and lipids on day 0 in these babies. But also, again, emphasizing that what we really want to be doing is getting breast milk into these babies. So we start oral colostrum at hour 0. We get the mums to hand express. We will be collecting that colostrum in a small syringe, and we will get the mothers to give their own babies their own breast milk within the first 12 hours of life. That starts the whole process. It's very good for the mothers psychologically. It's great for the babies. So yes, PN from birth, but also let's think about colostrum in the mouth as well.
Great. Next question. Any health consequences on cognition if choline levels are, I guess, too high, are high?
Yeah, I mean, choline is a very important nutrient. The metabolism of it is incredibly complex, and there are data in adults showing associations between certain choline metabolites and worse cardiovascular health. So all of our nutrients, we need to be aware that just giving more and more and more isn't necessarily good. I mean, proteins are really important, but only up to a certain point. I mean, if you give way more protein than the babies need, you will make the situation worse. So there's a therapeutic window for every nutrient, and that certainly applies to choline. Choline, when it's ingested enterally, is metabolized by certain taxa of bacteria into something called trimethylamine oxide, and that can be damaging to the liver. So I think when we start doing studies looking at high levels of choline, we do need to be aware of the potential kind of metabolic consequences. I think at the minute what we can see though is that the baby's choline intakes don't meet recommended levels. So I'm not suggesting that we're giving babies excess choline above what's recommended. I'm simply saying if we look at what we're doing at the minute, we're probably not meeting recommended intakes, and that is therefore an area where we could probably get better at it.
Great. We have time just for one question, and it's and this question is, well, maybe we already answered the difference between fresh breast milk versus pasteurized stored breast milk. We'll go to the last. How would you compare the development of executive function between preterm and the very low birth weight, I guess very low birth weight but term babies compared to preterm babies?
Yeah, again, very important to recognize that prematurity has different impacts from growth retardation. And a 2-kilo baby who is a growth retarded 40-week baby is very different from a 2-kilo 32-week baby. They've had different problems. they've had different deficiencies perhaps, and they have different outcomes in later life. So IUGR babies are not the same as preterm babies. So we need to know, particularly when we're talking about macronutrients, that there is no benefit from term babies receiving lots of extra macronutrients. There's no benefit to giving a term baby, even a 2 kilo term baby, giving them 4 grams of protein. It's not going to help them, and it may actually make them worse. Whereas there may be benefits for small babies. So most of your vulnerability comes from your gestation rather than your weight. You're always better off being more mature as opposed to being heavier. Clearly, we want babies to be a good weight and a good gestation. But gestation really has the biggest impact. But even term babies who are IUGR do have differences in their metabolism and cognition in later life.
Great. Well said, Professor Nick. So thank you very much. Unfortunately, that's all the time we have for today's discussion. Thank you, Professor Nick, for spending time with us this afternoon. And for
thank you very much, and lovely to meet you all, and I hope to meet you all in person one day when times are better. Yeah, thank you again.
Great. Professor Nick, do you have any parting message for the audience members?
Yeah, I guess my parting message is that all of us can do a better job of nutrition. It's really not that complicated. You don't need to understand the metabolism of choline to realize that getting breast milk into babies is a good thing. So I think my parting message is just to encourage all of you as clinicians to believe that you can actually make a difference if you want to do that, and just focus on the really simple things. Mum's breast milk and macronutrients, that's where I would start.
Great. I guess still breast milk is best for babies.
Absolutely.
Okay, dear doctor.